So smartphones nowadays are devices that are commonly used by almost everyone, in which they are the only thing that are being transported by us, but they're not being washed away, and therefore they are an optimum media to transfer bacteria. Therefore, several companies have produced antibacterial protection films in order to prevent the growth of bacteria. However, since the growth of bacteria cannot be seen by the human eye, its effectiveness is questionable. Therefore, for my experiment, I have decided to investigate the effectiveness of the antibacterial protection sheet, in which I decided to investigate coliforms, as coliforms are a marker of human contamination, and they are often found in food industries. And they can be found almost everywhere, but mainly in fecal materials, and they can also lead to potential waterborne diseases. So as the mechanism of the protection film I have decided to use, it is the Hydro AG Plus antibacterial liquid crystal protection film. And this protection film uses silver ions to prevent the growth of bacteria by depositing itself in the vacuole and cell wall as granules. And so the membrane will be damaged and exhibit structural abnormalities. I have decided to use petrifilms in order to look at the growth of bacteria, in which I use the 3M petrifilm coliform count plates, which are often used for environmental experiments. This method will be more efficient compared to traditional agar methods, as they are more time efficient. They do not require to remake the agars, and they do not require the agar dishes. They are also sealed, and therefore they will prevent contamination to be spreading. In order to make the petrifilm, sterile saline solution was prepared and placed on the petrifilm with 1 milliliter of micropipette. Then a spreader was used to avoid air bubbles, and it was left 1 hour to gellify. So the method of the overall experiment will be explained. I investigated survival of bacteria on a protection film with and without antibacterial properties, and the control, which was a plain surface, which in this case was the tablet. So the water was contaminated using coliforms. Then a cloth was placed on the tablet, which was contaminated with that water. Then the petrifilm were placed on the surfaces with and without antibacterial sheets and the control. Then they were incubated for 24 hours at 35 degrees Celsius. So my hypothesis for this experiment is that the antibacterial protection film will inhibit the most growth of bacteria, as it has antibacterial properties, and the least will be the control, because it does not inhibit any antibacterial properties. The results vary for the experiments, but in order to show some traits, this first petrifilm shows a petrifilm incubated without any growth of bacteria, and the second one shows a petrifilm with really numerous amount of growth. So the result of the overall experiment showed that the protection film with antibacterial properties actually had the greatest growth of bacteria, followed by the protection film without antibacterial properties, and the least was by the surface without protection film. This result, therefore, was against my hypothesis. And so, as a conclusion, the greatest bacterial growth was in the antibacterial sheet, and the control was the most efficient. As an evaluation, these results can be deduced from several factors, in which the first one could be from the oleophobic coating. So for the tablet I have used was an Apple product, in which when the patent was looked at, it has shown that it had some oleophobic coating, which prevented water to being deposited on the surface. And so this lack of water could have prevented the growth of bacteria. Also, these petrifilms, in order to form colonies, they need to produce acid. And so a deeper color will lead to a greater quantification. However, when I looked at my overall result, I have found out that the color of the bacteria was getting darker under control and the lightest on the antibacterial properties, which shows to match my hypothesis, and therefore have concluded that for the colony counts of the coliforms, the antibacterial property 1 was greater. However, overall, there could have been more bacteria under control, but it was not seen as they're not coliforms. Therefore, for future experiment, use of different petrifilms could be used in order to investigate different types of bacteria and their growth on the surfaces. Also, agar could also be used as it allows the growth of 48 hours. Since this petrifilm was designed to only be used for 24 hours, limited growth could have led to limited amount of results, and therefore using an agar of 48 hours could lead to greater results. A quick swab is also an alternative way to investigate environmental samples. And also, for future experiment, foam embedded with antibacterial surfaces could be used in order to investigate their growth and the survival of the bacteria.